Fertilization is a regulated process that occurs in ampulla of the oviduct by which highly specialized male and female gametes unite to form an unspecialized totipotent zygote. Let us break down this definition. So this is a process which is complex and coordinated. It also tells us about the site where it is happening. It is happening in ampulla of the oviduct. There are two specialized cells involved in this. These are the male and female gametes, the sperm and the ovum respectively. And the result of their fusion is a zygote which is capable of producing all the cells, tissues and organs of an individual, totipotent. Let us see how this fertilization is accomplished. The background that we have is a male gamete, sperm, and a female gamete, oocyte. The oocyte is viable for a period of 12 to 24 hours, whereas the sperm is viable for about 24 to 72 hours. So, if fertilization has to happen, the coitus must take place no more than 3 days before the ovulation till 24 hours after the ovulation. If this episode has to stand a chance of conception, then in that given time window, coitus has to happen. Let us now talk about the oocyte transport towards the site of fertilization, after which we'll speak about the sperm transport to the site of fertilization. First, the oocyte. Shortly after ovulation, fimbria of the fallopian tubes sweep over the surface of ovary, and upon ovulation, the oocyte with some granulosa cells is carried in the tube by the fimbriae due to rhythmical contractions of the uterine tubes. Further transportation is done by the action of cilia that are present in the lining epithelium, which is columnar epithelium in the fallopian tube. And that is how the egg reaches the site of fertilization, that is ampulla of the oviduct. Now, what is the fate of this egg or the oocyte that we are talking about? It can either get fertilized, or if it does, it will be transported to the uterus in the form of a conceptus, and upon reaching the uterus, it will implant and grow there. And if it does not get fertilized, yet it is transported to the uterus, and in the uterus, it is going to be resorbed. Let us now see what happens to the sperm. The 200 to 300 million sperms that are deposited in the female genital tract, that is vagina, at the time of coitus, many of them leak out of the vagina immediately after deposition. Out of the others which are remaining, some are destroyed by acidic vaginal environment. Those which survive try to swim towards the site of fertilization, but eventually fail to make it through the cervix because of the narrow cervical canal and the characteristic of the cervical mucus. And those we actually enter the uterine cavity, they are dispersed in the uterine cavity or are destroyed by the phagocytic leukocytes. So a very few of the sperms actually succeed in reaching the uterine tubes and try to get to the site of fertilization. Just by being in the fallopian tube does not guarantee a sperm that it can fertilize the ovum, because one very important process is still remaining, and that is the process of capacitation. Sperm must undergo capacitation before they can penetrate the oocyte, and this process of capacitation takes approximately 7 hours. Now, those sperms which are there in the female genital tract and are capacitated, they find a guiding cue of sorts coming from the oocyte. There is yet unnamed sperm chemotropic factor which is secreted by oocyte and the surrounding follicular cells, which actually guides the capacitated sperm towards the oocyte in a chemotaxis process fashion. So it will not be wrong to say the sperm finds the human egg attractive. So the gamete transport towards the site of fertilization now brings us to what are the barriers for fertilization. Well, we have first barrier in the form of corona radiata. The second one is their zona pellucida, and third is the oocyte membrane. These three barriers will be conquered and eventually fertilization will happen. For that process of fertilization to reach its logical conclusion, it has to pass through three phases. There are three phases to the fertilization. Phase 1 is penetration of corona radiata. Phase 2, penetration of zona pellucida. And phase 3, fusion of the cell membranes of the sperm and the oocyte. Let us see them one by one. The first phase is penetration of corona radiata. Capacitated sperms pass freely through the corona radiata cells due to three factors. First, there is some help by the hyaluronidase that is present in the seminal plasma, that is coming from the acrosome. The second factor that aids in this are the tubal enzymes present within the tubal secretions. And the third is the motility of the sperms themselves. After penetration of this corona radiata comes the penetration of zona pellucida, which is phase 2. Now this penetration of zona pellucida is not just a phase, but it involves triggers and cascading mechanisms to actually bring about certain changes necessary for the fertilization to continue as per plan. There are species-specific receptors present over the zona pellucida, ZP1, ZP2 and ZP3. Those are the abbreviations for the receptors. Out of them, the ZP3 is a species-specific receptor that binds the sperm, and through that binding, it mediates a very important reaction called the acrosome reaction. When there is fusion of spermatozoal plasma membrane with the outer acrosomal membrane, the acrosome reaction is triggered, and with that the contents of the acrosome are released. The contents of the acrosome include acrosin and a trypsin like substance. It is released at the place of contact and it allows the sperm to literally burrow through the zona pellucida. This exposes the equatorial segment of the egg, and that's the place from where the sperm is going to fuse and enter in the cytoplasm of the egg. The contact between the sperm and oocyte triggers calcium waves radiating along the oocyte surface, culminating in two more reactions, cortical reaction and zona reaction. So what is cortical reaction and how is it interrelated with the zona reaction? Cortical reaction speaks about cytoplasmic granules present in the peripheral cytoplasm of the oocyte. They are called cortical granules. They contain a mixture of enzymes including several proteases which diffuse into zona pellucida following exocytosis from the egg, and these proteases trigger the zona reaction in which there is alteration of structure of the zona pellucida in such a way that those receptors, species specific receptors which were there for binding to the sperms, they are inactivated so that no further sperm will be able to penetrate the zona pellucida, and that also makes sure that the oocyte membrane also gets impenetrable to other spermatozoa. So zona pellucida becomes impermeable, so does the oocyte membrane. This brings us to phase 3 of fertilization, which is fusion of oocyte and sperm membranes. When these two membranes fuse, the head and tail of the sperm gains inside the oocyte cytoplasm. The other parts stay behind. So what is the oocyte response to this sperm entry? The responses are: number one, triggering of cortical and zona reactions to prevent polyspermy. Number two, resumption and completion of second meiotic division, leading to formation of second polar body and a female pronucleus. And third, an important one, metabolic activation of the egg. That brings us to the final stop in the process of fertilization, fusion of pronuclei. The nuclei of the ovum and sperm swell to form the female pronucleus and the male pronucleus respectively. Each pronucleus replicates its DNA, the nuclear envelopes disappear, and soon they enter into a spindle formation, which eventually gets completed, and there is division of this single cell, giving rise to two cells, and it will no more be called a ovum or a mature egg, it will be called a zygote. The zygote is the one that immediately divides into two cells. So what are the effects of these neatly orchestrated process, the fertilization? Well, the effects are restoration of diploid number of chromosomes, results in variation of the human species through mingling of maternal and paternal chromosomes, determination of gender of the new individual. So whether it's going to be a male fetus or a female fetus is decided exactly at the time of fertilization itself. Next is initiation of cleavage division. Cleavage division is a series of mitotic divisions that give rise to increased number of cells. As more and more divisions happen, the numbers keep increasing. And metabolic activation of the egg, that is very necessary for the further process to continue as per expected plan. Well, that's fertilization for you. Happy learning.